colorimetric analysis. This is called DSC. Useful information. This study will give us the useful information about the possible interaction about compounds as shown in the figure. These DSC thermograms give peak at 110 degrees, whereas the pure gallic acid showed at 90 degrees. And the crystal melting temperatures around 266 degrees here. So on the other hand, the GaCSNP loaded particle give a quite different endothermic peak from the individual components. So this will suggest that such reduction in the melting point and lowering in the phase transition temperature, we can call it, gives information about the reduced crystallinity of the prepared gallic acid. Comparable results were observed in gallic acid phospholipid complex, where the crystal structure of gallic acid changes to amorphous in nature in chitosan nanoparticle, followed by XRD analysis. So this will give generally the physical structure of the prepared composite material of pure gallic acid as well as the gallic acid incorporated chitosan nanoparticles with the different peaks as shown here. So in contrast, the different diffraction is called as a diffraction of significant peak, showing the crystalline nature of the pure gallic acid could not be detected in this gallic acid incorporated chitosan nanoparticle. So it was concluded from the study that gallic acid is probably present in amorphous or disordered crystalline nature within the chitosan nanoparticle. So based on this, we further move to the Fourier transform infrared spectroscopy analysis, that is called FAR, of these different compounds CSNP, chitosan, and GaCNP. So these different analyses will give these different functional groups that will give idea where our prepared component is chemically stable or not. So a lot of chemistry has been involved. We don't want to talk much about this because these interactions reduce the chitosan solubility, are responsible for the separation of chitosan in the form of nanoparticles. This group is very important for us. It should not vary as compared with our individual group. So scanning electron microscopy has been done to confirm this size analysis, where we can able to see the yellow green color marked here within 200 nanometer, which indicates the transient. That means the nuclear transport during the in vitro analysis. Followed by the important analysis called as a DPPH assay. It is called a basic biochemical assay. It is used to quantify the antioxidant property. So what will happen? The principle of this method totally depends on the quenching of a single electron of DPPH by a compound having the antioxidant activity. Generally, the gallic acid is a natural plant polyphenol with high antioxidant activity, shows excellent scavenging property, even though when we use the concentration of 0.05 mg per ml. So in the case of gallic acid incorporated the final nanocomposite material, the highest scavenging activity was found to be 92.85 at 0.5 mg per ml. It was observed that DPPH radical activity linearly depends on the percentage of encapsulation of gallic acid in this total incorporated chitosan nanoparticle. Next study, we use the Nin-nitrine assay. So why we use this Nin-nitrine assay for the physiochemical characterization is it is a simple biochemical assay used to determine the cross-linking efficiency, where we use the genipine. This is the plant-based T component that has been commercially available, used as a natural cross-linker as compared with any chemical cross-linker showing some cytotoxicity. So you use the genipine, a natural-based cross-linker, to cross-link collagen as well as the fibrin. So what we observe is the degree of cross-linking, that is the amount of remaining free amines, was qualified through Nin-nitrine assay, showed at 0.3, 0.5, and 1 nanometer. So this will help to determine the cross-linking degree of the scaffold, and there we can able to see the 66.78% with 1 mm concentration of genipine. So these results confirms that cross-linking happening because of the oxidation reaction between active chemical group, which increases the mechanical stability of the prepared polymer material. So this is the overall collagen fibrin scaffold have been looking like that. It's like a spongy cotton-like substances, different size, shapes, and whatever the purpose we can able to convert according to our experimental analysis. So the overall scaffolds has been done both transition electron microscopy, scanning electron microscopy to show the particle size analysis. As I told initially, we have done previously with the individual components. Once we make it as a fibrin scaffold with the collagen, fibrin, and the collagen alone, we have done a pair analysis to make sure the functional groups have been properly present. One of the important in vitro analysis we have done in porosity and swelling. These two important parameters play important role when we do in vitro analysis. So first I will talk about the porosity. Porosity and pore size of the scaffold have direct implication on the functionality in vivo as well as the in vitro condition. So the porous nature, what will happen? The porous nature of the scaffold allows the gaseous nutrient exchange on the wound site, which is essential for the cell proliferation and vascularization, thereby facilitating the progress of healing. It also offers the mechanical interlocking between the scaffolds and surrounding tissues to improve the physical stability of the scaffold and also enable the effective release of the bioactive compounds at the wound site. So, followed by the swelling analysis we carried out between non-cross-linked scaffold and cross-linked scaffold. So, what will happen? The swelling ratio defines the fluid uptake ability or capacity of the scaffolding material, plays significant role in the absorption of body fluids, excess blood, exudate present at the wound site when we do the any in vivo analysis. So, this will also help for the transfer of cellular nutrients and metabolites inside the scaffolds. So the swelling capacity of the scaffolds got decreased after molecular cross-linking with genipin. So it indicates that there's a direct relationship with the structural stability of the scaffolds when we use the in vitro and in vivo application. So the itophilic materials such as collagen have poor mechanical property and its porous structure collapses when kept in hydrated environment. So based on this swelling analysis, we further move to the thermogravitic analysis of the prepared scaffold. So this thermogravity is a simple and reliable method to study the polymer degradation with starting from 0 to and maximum 800 to 900 degree. So the higher transition temperature suggests the scaffold having the high stability at high temperature environment, then compared with a pure collagen. And thermostability also controls the durability of the scaffolds. So in vitro biodegradation we have done at by using a lysozyme, that enzyme that has been present in our system. So comparing with the non-cross-linked scaffold, the cross-linked scaffold leads to the sustained degradation, which could be very helpful for the material preparation. Followed by the in vitro drug release profile that also be carried out between 0 to 100 hours. We can able to see the sustained drug release in the comparing with the gallic acid CSNT scaffold as compared with the collagen fibril scaffold. And also we done the similar DPPH assay both in the scaffolds containing collagen fibril scaffold and the transphenol polyphenol gallic acid containing collagen fibril scaffold. So showing the better efficiency of a scavenging activity. So coming to the in vitro study. What we have done in this in vitro study is specifically we used 3T3 skin fibroblast cells with a different concentration of 24, 48 and 72 hours time period ranging from 20 to 100 micromolar of this gallic acid has been treated, which could not found any significant toxicity. Followed by the viability estimation of the conditioned medium has been done to show the better stability of the prepared or biocompatibility of the prepared material on these 3T3 fibroblast cells. Similarly, the live and the dead staining has been carried out by using calcium AN, calcium hydroxymethyl ester derivative, followed by the counter staining with the propylium iodide with different scaffolds containing collagen fibrine and gallic acid containing collagen fibrine scaffold. We can be able to see there is no dead cells, much red cells, which indicate the dead nuclei, and we can say be able to stain most viable cells on these 3T3 fibroblast cells. Followed by the basic assay is called the cell migration assay. In control, cold fibrine scaffold and anacomposite scaffold, where IW is the initial wound, where 0th hour we can able to see all the initial wound is at the equal level. Followed by the 24th hour, we can able to see the complete healing in the nanocomposite scaffold, and 48 hours it has been completely healed, and the migration has been happening properly as compared with the control. So, based on these in vitro data, we move to the preclinical study. We used a Vistar albino rats model where we anesthetized these animals and we created 2 × 2 centimeter wound excision wound model. So here, this is the control on day 0, collagen fibrine scaffold control model, and the gallic acid plant paniculol containing titosan nanocomposite scaffold. So if you see day 4, day 12, and day 6, at the end of the day 12 itself, there is a complete healing of the gallic acid incorporated titosan nanophyte scaffold, and the percentage of wound contraction has been calculated. We can able to see nearly 80 to 90% of the healing has been happening in day 12 and complete healing happening in the day 60 as compared with the control group. So this suggests the prepared nanocomposite scaffold having the better efficiency. So important component of this period of reepithelialization. So this percentage of period of reepithelialization has been calculated from after the sacrificing these animals, where we can able to show that The importance of the re-establishment of epidermal surface act as a permeability barrier for excessive water loss and also prevents exposure to the microbial infection. So if you see the period of epithelialization in control rats were bandaged with a sterile gauze showing around 25 or 24 days, and that a rat treated with the collagen fibrin scaffold was 20 days, and whereas in case of gallic acid infiltrated chitosan nanoparticles showing nearly 16 days. So this suggests that there is a complete wound closure with a reduced period of re-epithelialization on the day 16. And two important parameters which I am going to talk now detail about is the hydroxyproline as well as the exosomes. So to go detail about what is I told initially itself, collagen is a predominant protein component present in the ECM, plays a vital role in all the stages of the wound healing. So during the wound healing cascade, there is a swift increase in the synthesis of collagen by the fibroblast cells, which provides structural stability and integrity to the newly formed tissue at the wound site of the area of the wound healing is determined mainly by the natural process called de novo collagen deposition and maturation. So we cannot directly measure the collagen. The indirect way to measure is the hydroxyproline estimation. So this will serve as a key factor to determine the amount of collagen that has been present in the granulation tissue. So we collected on the different days 4 and 8 and 12. We can able to see the granulation tissue formation is a much early phase as we discussed in the introduction. This is the important place. After day 8, it starts decreasing in all the groups. So this compare that prepared nanocomposite scaffold has the ability to form the ECM formation. Another important biochemical content is called exosomes. It is called as a matrix molecule that acts as a ground substance, supports the synthesis of the new ECM. These exosomes helps to stabilize the newly synthesized collagen fibers by enhancing the electrostatic and ionic interaction with it. It is reported that exosome content was remarkably increased during the early stages of the healing cascade, as shown in the figure. We can able to see rats treated with the gallic acid containing nanoparticles showed significant increased level of exosome, followed but decreased compared with all the collagen and collagen fibrin groups, indicating the faster wound healing process. So this is a very basic histological study where the mass and trichome staining we carried out, where the control we could not able to see the excessive blue bundles, which indicates the collagen formation. Whereas in the treatment of nanocomposite scaffold, more collagen or ECM formation has been happening. That has been indicated with the arrow mark with the C as compared with the control. Whereas in hematoxylin using staining has been shown to show the new dermal formation as well as the epidermis formation has been indicated in the black arrow as compared with the control. So this is the control model, normal wound model we have done. Now I'm showing how we have created the diabetes by using a chemically available compound called streptozine. We used 110 mg per kg body weight and 60 milligram per kg body weight of streptozine in cold 0.1 citrate buffer. We used as a drug delivery agent. Then pain weight has been monitored. Once the glucose reaches after 2 weeks, the wound was created. And similar experiments we have done. So if you see comparing with the control and the normal wound experiment we have done, we have extended this study because the normal comparing with the normal wound, the diabetic wound will take much longer time. So we extend the prolonged time between day 12, day 16, and day 20. We can able to see at least as compared with the day 12 in normal wound, it will take nearly day 16 as compared with the control and the collagen fibrin scaffold, the GACNP, that is the plant polyphenol incorporated scaffold, completely healed on the day 16. Similarly, wound contraction measure. So as I told previously, in a similar experiment has been carried out to show the period of epithelialization, indirect measurement of hydroxyproline estimation and exosome. Both the 8 days in the diabetic model has been increased significantly, which able to form the ECM membrane. Similarly, to support these, we have done further the mass and trichome staining and hematoxylin staining for the new blood cells formation as well as the collagen tissue formation. So this chapter, I want to summarize these. The present scaffold containing the collagen fibrin infused with the gallic acid containing chitosan nanoparticles has the potential properties of wound dressing materials and could be beneficial for faster re-epithelialization and wound contraction. Now, the last chapter which I'm going to talk in next 5-10 minutes is the pricing. It is a plant-based polyphenol loaded with alginate chitosan composite material for a normal as well as the diabetic. So the idea between this chapter and the previous chapter is, we used a nanoparticle-based drug delivery. Here we used a simple sol gel method with the available material of pricing as well as the alginate-based material. with chitosan for this diabetic and normal model. So what is chrysin? The flavonoid compound mainly found in honey, propolis, mint, and blue passion flower. It has several biological functions. So we showed this is the first experimental investigation of chrysin loaded with the sodium alginate and chitosan. It forms a porous 3D scaffold that can be used as a topical dressing material. So we hypothesize that combining all three components, chrysin loaded with sodium alginate and chitosan, offers a synergistic outcome to accelerate the natural process of the healing. This is the overall scheme. How we mix the chitosan solution with the sodium alginate is the polysaccharide, then the chrysin polyphenol homogenized, and simply we make it as a salt gel method as a spongy substances, and detailed chemical characterization in vitro and in vivo have been done. So this is some of the porosity analysis. Just I want to show very fast because these have been very simple. 1:1, 1:2, 1:3. We can able to see at 1:3 the better porosity, followed by the swelling analysis. We can able to see the 1:3, the green color one showing the better swelling, and biodegradation at the 1:3 is very sustainable up to 48 hours. After that, it got degraded. Similarly, these pore size of the scanning electron microscopy at 1:3 showing the 200 to 400 micromolar pore size in the drug loaded scaffold. So this is the detailed FTR analysis has been shown with the different functional components that has been present in the each material be prepared, chitosan, alginate, conjugate chitosan, as well as the alginate chitosan chrysin. So these will give the successful incorporation of the chrysin in the prepared scaffold of the different functional groups. Similarly, XRD, TG analysis and DSE and drug release further substantiate our prepared scaffold is biocompatible and can be used for the further in vitro analysis. So this is important analysis we have done in biological interaction between the scaffold and hemocytes, where we used the red blood cells isolation, where the RBC morphology was observed between the two different scaffolds to show the interaction between the scaffolds and the red blood cells. So the main objective of this study is that the degree of protonation of the amino groups of chitosan generally present on the red blood cells play a significant role in the absorption. So the scaffolds are fabricated from the alginate generally produced by chemical bridging of addition of a calcium ion and rapidly releases these ions in exchange for sodium ion in contact with the blood. Alginate will possess the hemostatic property that plays in when we incorporate it in any animal system or in our body. Therefore, alginate has been popular in many biomedical applications. One is the wound healing. However, after the drug incorporation, it was observed that attachment of these blood vessels was equivalent to alginate chitosan chrysin scaffold or compatible with the blood cells, and incorporated plant-based polyphenol chrysin did not show any significant toxicity, suggesting that these alginate chitosan chrysin scaffold positively influences the physiological action during the coagulation process. So the vice versa, the result will come the BCL gauge, the control we show the better higher BCL value, whereas our plant incorporated chitosan chrysin showing the lower blood clotting index value. Similarly, the NH53 mouse fibroblast cells were being used with a different concentration, and we can able to see up to 20 to 30 micromolar concentration of these chrysin did not show any toxicity. Similarly, the cell viability showing that better biocompatibility. Further, by live and death staining has been carried out in control as well as the alginate chrysin scaffold and alginate containing chrysin chitosan scaffold, showing that there is no red cells. That indicates that there is no red cells on this PA staining as compared with the control. Similarly, scratch assay confirms the prepared scaffold is the better wound healing potential. So like that, we have done a similar experiments where we can able to see on the 12th day there is a complete image healing of these wound healing process happening as compared with the control group here, where we can able to see the chitosan chrysin alginate showing the better healing potential. These are the supporting evidence which I showed detailed in previous slides or previous chapters where we done similar biochemical analysis, hydroxyproline and exosomes. Further to substantiate, we have done a Mason trichome staining and hematoxylin staining. Similarly, the diabetics model has been created where we have done up to day 16. We can able to see nearly, we can see 85 to 90% have been healed completely, whereas day 20 is healed completely 100%. Whereas in the case of control group, it takes only day 12. Similarly, the period of epithelialization, hydroxyproline estimation, and exosome estimation has been carried out. And further, it has been substantiated by using a collagen staining, by using Mason trichome, where we can able to see the excessive amount of collagen formation has been happening in these plant polyphenol incorporated pricing scaffold as compared with the control group. So this the chapter I want to summarize that as I told in previously all the results, the one line I want to show here, these studies observations suggest that these alginate chrysin chitosan scaffold can be used for dermal wound healing and regeneration application. So what could be the take-home message from this presentation? I just want to summarize. There are several techniques with various combination of natural and synthetic graft materials that can be utilized for achieving any wound or bone tissue regeneration. So there is no single ideal technique or graft material to choose in clinical practice. So what we can expect that the biomaterial which 3D architecture combined with any growth factors or any incorporation of allogenic cells will demonstrate the vast improvement in the graft and biological tissue interfacing based on the knowledge gained from recent research and allow the clinicians to achieve more predictable clinical results with regards to wound regeneration. So the treatment protocols that involve the cost-effective, more reproducible and less technique-sensitive procedures and biomaterials need the constant revisions in upcoming years in light of new development of tissue regeneration therapeutic aspects. Last but not least, I would like to thank my research group, my present research group, and she is the candidate, Mrs. Pallavi Shamkapakar. Now she is a Dr. Pallavi Shamkapakar. She is the one responsible to carry out all these wound healing experiments in our lab, and all my present PhD students and project fellows. And I'd like to thank all my funding bodies from DST, SERB, CSCR, FTT, and FDR. So all these fundings can able to make a better research in our lab. Thank you for all your patience, and I am happy to hear any questions from all of you.